So this afternoon we're going to kind of continue our conversation of a better understanding of STXBP1 disorders, and our first speaker is going to be Wendy Chung. I'm going to make sure I get this title right. Wendy's the chair of the Department of Pediatrics at Boston Children's, and she's also the principal investigator for the Simon Searchlight study. STXBP1 has been part of this study for many years, and Dr. Chung's going to give us some insights into that study this afternoon. So give her a warm welcome.
Thanks, everyone. I'm thrilled to be with you here today, and I was saying to Charlene at lunch, I'm going to pull an audible, and although my slides won't necessarily reflect everything I'm going to say, I don't want this to be duplicative, and I want to give you some insight into some things going forward. So let's see, hopefully my slides are up and running. Anyway, as we're trying to advance the slides, what I'll say is that Simon Searchlight has now been going on for about 15 years, so just a little bit less time than we've known about STXBP1. As we've done this, it's an umbrella, as the way I think about it, across neurodevelopmental genetic conditions. And within doing this, it's global, and we've tried to really de-risk this and make this a resource that makes it an amplifier for what families do from around the world to be able to increase their impact. So this is sponsored and funded solely by the Simons Foundation. It has been since its beginning, and it will continue to be. And with their generous support, we've been able to do many things in partnership with other groups. And as we do this, we try and do it responsibly. That is to make the information de-identified, so no one knows who anyone is, but be able to make it easy for researchers, whether they're in a company, whether they're in a university, wherever they might be around the world to do this, to be able to make the most of your contribution to research. And part of what I'm here today is to give you an update in terms of what's been going on, and to listen in terms of what you as a community need going forward so that we can position ourselves to be able to help you best. So as I was alluding to, and thank you for those of you who have seen Gabi out in the entryway and contributed samples, blood samples to Combined Brain to be able to power this. As we've been doing this, there are over 130 publications so far across the Simon Searchlight conditions. And one of the beautiful things about this is it's not just STXBP1 in isolation, but it's the ability for many researchers to look across conditions and to be able to see how one condition fits into the ecosystem overall. So that's a relatively unique way of being able to do things, and one of the features that I think many of the investigators have used. As we do this, part of the goal is to translate this back into things that are meaningful for your daily care and for your information, for your daily lives. And one of the ways we do this, we believe very much in open data science, making sure the information that's published that comes out is freely available to anyone in the community. And one of the easy ways to do this is through something called gene reviews, to be able to make sure that gets updated in a very sort of quick way that your clinicians, your doctors, when they're seeing you, can be able to go to that website, see up-to-date information, and be able to process what might be new information to them, but very quickly in real time in a way that helps you going forward. That also helps in terms of eventually getting services covered, like insurance, in terms of different recommendations, and it builds the evidence as we go forward. So again, coming out of this, our original, this is just the count in terms of the number of papers that have come out of this for STXBP1, and in addition, we have, through the Simons Foundation, funds that come to this family meeting, as an example, as well as to the investigators who are doing the research. So it's not simply just you putting in information, but it's also funding some of the researchers to be able to use the information for you. So let me give you a brief update then in terms of what we've been learning, and to say that I do think it's changing, and this is actually, to me, dynamic and important, that over time, I think the natural history is changing. So in other words, what we're seeing, and I think that's actually, for me, an opportunity and signifies hope in terms of what we can be doing for the future. And I'm going to put in a plug for that in just a second. So as we do this, we, I think, do a very good job of making sure that people in the registry really truly have verified diagnosis in terms of STXBP1. I will say it doesn't happen very often, but I'm a geneticist and I actually review the genetic reports in some cases. And every once in a while we have someone who thinks that's their condition, but it's actually not, and we help them get to their correct diagnosis. And this is very helpful in terms of the researchers that use the data being very sure that these are in fact the data that they want to. Individuals, as they're going through the process, I'll emphasize this is not a one and done. This is, as you've been hearing about, really a living, breathing longitudinal registry. And the beauty of this is it's all online. So no matter where anyone is in the world, they don't have to be able to go to one of the clinical sites, as you saw from the beautiful natural history data earlier. And that's important globally, especially. So people can be anywhere in the world and still contribute. And it's synergistic, additive to what's going on with the in-person assessments. As we're doing this, another portion of this is the blood sample that comes in, and I'll get to that in a second. Obviously, that's something that we do at in-person family meetings, but we also have services for people to be able to do it from their homes and send that in. And again, this is something that's living, and every year we continue to fill in the data. I'm not going to talk too much about this because you've already heard about this and you're going to hear about this next, but I will emphasize that this is a stick plot. It's not very fancy, unlike some of the three-dimensional plots you've seen earlier, but this is that lollipop plot you heard Ingo talk about before in terms of all the different genetic variations. And the ones I'm showing on the lollipop here are single amino acids or single building blocks in the protein that get swapped out. And as you've heard about, it is interesting that certain of these positions, the numbers in the parentheses, means that there's more than one person with the same genetic variation, and everyone isn't represented here. This is just the people who are in Simon's Searchlight. But there is, and again, I won't go deep into the biology, but I don't think all the mutations are acting by the same mechanism. You've heard this a little bit today, but there's a deeper dive in terms of that, what we call genotype-phenotype correlation that you heard that many times we and the genetic counselors get in advance, and it may actually have an implication for therapeutic strategies going forward. So to me, it's an important area, an opportunity going forward. In addition to this, but what I haven't shown is that there are all sorts, this is that gray box in the upper left, there are a bunch of people who have, we think, functionally a copy of the gene that just doesn't show up to work. So what I mean by that is it has a premature termination, it has a deletion, but something so that you've got half the amount, haplo insufficiency, half the amount of protein. And for that, it makes sense in terms of being able to restore the function, give that one copy back in some way. Whereas with the missense variants, like I said, it could be more complicated. And I'll let others talk about that are much more knowledgeable than I am. In doing this, we have an international, as I said, group, but we do have a huge number of people here in the United States. I will say, and I get this perspective simply because I can see across conditions, you are very lucky. You're very lucky that the leadership you have in this community in particular is extraordinary. And I will say that in terms of across, I see a lot of conditions and rare diseases, not just in terms of brain conditions, but across everything, literally hundreds of them, and you are really uniquely positioned in terms of having leaders that have been very thoughtful and strategic in terms of moving you forward. So I'll just put that out there. In doing this, it is, as I said, a global condition, and there's no reason to think that it should be any one place more than another. So even though I'm showing you that we have a large number of people in the United States, just to set the sort of, put it in your head, it's not because this is an American condition. Really, this is, we think, equal opportunity around the world, but it simply reflects access to genetic testing. We were actually talking about this over lunch in terms of some of the other things, but I'll show you the age distribution as well, and that's skewed. I don't think it's just what we see. I think there are many more people out there. This is what I was alluding to in terms of the different cell lines. And again, the Simons Foundation has been maintaining this repository that, again, is available to researchers in terms of being able to use this. And importantly, to make these induced pluripotential stem cells or brain cells in a dish or even organoids or mini brains to be able to test out before they go into people. This is a resource, and it's important that it's not just one person that's represented here. It's actually many different individuals with many different genetic variations. And with this, as I said, it's a very nominal, literally the cost of shipping in terms of researchers being able to do this. And so this is what's going on behind the scenes in terms of the blood samples that you've been donating. With this, the Simons Foundation continues to both distribute as well as expand this. And each one of these lines that's made costs about $15,000. So this is no small investment in terms of powering your community. And these are the second list of variants. So within this, what are the data we see, and I'll put out the limitations of what we see, because there's what we see, and what I worry about is what we don't see. So as some folks have already pointed out, we're skewed in terms of who's accessing genetic testing in this era. And based on who's accessing genetic testing, we're largely seeing what I think is the beginning of the lifetime journey. So we're seeing individuals really, almost exclusively, not quite, but almost exclusively, who got their diagnosis as children. We're watching them age up. So based on the fact that we've known about this for a while, some people are now adults. But what you're not seeing here is you're not seeing individuals largely who are in their 40s, 50s, 60s with this condition. Now, the question is, where are they? Or do we have them? I'm sure they're out there. I'm absolutely positive, because I myself have done things where I've tapped into some of our adult communities to know that our conditions are in those communities, but they're not accessing the genetic testing. And I'm not blaming them. I'm not saying they have to, but just to say that I don't have a lot of great long-term outcome data, because we simply don't know all of those people, and they're not represented in the registry. As we do this, not surprisingly, we're almost equal male-female, and this particular snapshot that I'm showing you represents 133 individuals. Again, less than, you've got an overlapping, but not exactly the same as what are in the natural history study that you've seen already today. I'm going to put in a little plug for something that's coming, because you're going to have some people coming into your community that are coming in through one of my, I call it a sister study. So I've got another study that I'm doing called the Guardian Study, which is called Genomic Uniform Screening Against Rare Diseases in All Newborns. And in that particular study, the point that I want to make for you all is we're identifying those babies, those little tiny babies that are just a few weeks old before they have symptoms, and identifying some of these genetic conditions like STXBP1 and encouraging them to join your communities then after they receive the diagnosis. I tell you this because I actually spent this morning with one of them online. They're coming in and they don't know what to expect. Unlike you all who came in at least knowing something about what was going on, they're coming in as a blank slate, not knowing what to expect, and they're looking to you all for guidance. And as we're doing this, I really truly believe this is the case, because clinically, I still see patients every Monday. I do know that my younger children, I think, are doing better than my older patients that have grown up with the condition, where we didn't have some of the tips and tricks that I think we've all learned together. And so I think you've got very valuable information that I know you're sharing with each other, and that I hope you're starting to share with the next generation of children growing up as well, because I do think there are more things that we can learn from doing this to make the next generation better. And clinical trials and the genetic therapies for sure are one of the important ways going forward. But even without that, I think there are other things that we're learning that we want to be able to implement as soon as possible. And part of the justification and the reason that I have this other study going is to be able to identify these individuals before the epilepsy starts, with the hope that if we get that under control, that we may be able to have additional possibilities in terms of brain development for them. So largely, I'm not going to go into most of the neurological manifestations, because other speakers have done that even better than I would do here. But just to say, not surprisingly, this is largely a condition that's affecting the brain, and to the extent the brain sends nerve cells to other parts of the body, but there are various different diagnoses. I'm going to focus instead on a few of the medical issues, and I want to reassure you that largely there are not medical issues coming on. So not surprisingly, this is something that's primarily affecting the brain, and to the extent the brain affects the body, that's what the effect is. But what we're not seeing, and I look for very, very carefully, is we're not seeing things like cancer. We're not seeing an increased risk of diabetes. We're not seeing an increased risk of heart conditions. We're not seeing an increased risk of kidney disease and other things. We didn't see a huge increased risk with COVID in terms of other things, any more so than we saw with kids that are medically fragile. So granted, we have that as a baseline, but we're not seeing any additional problems with the immune system or the way the immune system's working. So that's the good news. Not that life wasn't complicated enough already, but I'm not going to add to your plate in terms of other complexities going on. So what are some of the things that it might not be obvious how those things connect, but we do have the brain cells that are affecting, for instance, the gastrointestinal tract. So I do think it's largely through those connections that we see problems like constipation, reflux, to a certain extent problems with motility that we see. But I don't think it's primarily an anatomical problem with the way the intestine is structured. I think it's really a functional effect. We are seeing some issues when it comes, for instance, to orthopedic issues, which again, I don't think are primarily that the bones have a developmental bone problem. Other conditions I know do have that. I don't think that's this. I think it's the muscle tone problems that we see that are having an effect on how strong the body is and whether or not we see scoliosis or curvature of the spine or what the muscle tone is that may cause problems. But I don't think fundamentally we're seeing a problem with the bones themselves. In a similar way, sometimes we can see problems with growth, but again, that may have more to do with feeding and nutrition in terms of how much the kids are feeding rather than primarily a pituitary or primarily an endocrine problem with growth hormone deficiency. Vision, you've heard a little bit already. Some of the visual problems I think are really in the brain, so problems that we have with cortical visual impairment in terms of that, but we also have some problems with the eyes in terms of innervation to the eyes, the muscle tone in terms of the eye movement. We are seeing some of those issues. But a good news, again, to the extent that I can give you good news today, I don't want you to worry about any additional things. Again, these are some of the neurological findings. I'm not going to go into those in addition because you've heard about those already today, but largely the neurological findings have been treated medically with medicines, occasionally things like a vagal nerve stimulator, but haven't required a lot of surgeries. And in general, I would say the amount of surgery has not been excessive once we get out of the first couple years of life. Seizures you've heard about, so I'm going to flip through those quickly, and you've heard about the course in terms of the seizures over the life course and what we're still learning in terms of puberty. For the eyes, as I was alluding to a little bit, I think the eyes to me are important because it's the window in to get information in, to be able to take the world in around you, to be able to know literally what's coming at you, and to be able to learn and interact with the world. So I do think this is a very important thing to be able to do as much as we can for vision, but I realize also how hard it is to be able to assess vision. And I think most of you are probably going to a pediatric ophthalmologist, but a pediatric ophthalmologist who specializes in children with developmental disorders, even better if you can manage to do that, because there are tips and tricks that they can use to be able to even just get a good eye exam and to be able to see what's going on. So far, I'm not seeing problems with the retina itself. And I say this because there can be some eye conditions where we have to do very specific eye exams because there are problems with the retina. I am not seeing that here. So I don't think we're going to have to do any special types of things. We'd have to put the kids under anesthesia or do anything like that to be able, it's just a standard good eye exam, but with a pediatric ophthalmologist, preferably with developmental disabilities experience. The main thing in terms of surgery is that we are seeing as the kids are going through puberty and they're going through their growth spurt, a little bit more scoliosis, a little bit more curvature of the spine that we're seeing. So it is one thing to watch for. We're not seeing a huge amount of kids that need to have surgery or rods in their spine to be able to help with this, but that is the one thing that changes over the life course, and we'll see as we have more kids going through puberty. As I said, in terms of endocrine, a little bit in terms of growth, not massive problems, but a little bit that we're seeing in terms of growth issues. And again, with the heart, I would say these are minor things. Sometimes a little bit of a hole in the heart, sometimes it closes on its own with a septal defect, many benign murmurs that don't need anything. But again, we'll see as the kids are growing up if any of the sort of adult types of heart conditions start coming up. Gastrointestinal issues are the thing that we see most commonly in terms of other parts of the body. For many of you, this can be used with a combination of medications for reflux, medication for constipation, and dietary adjustments. And many times, once you get it right, you can sort of control things a little bit better. But I know until you get to that point, sometimes it can be problematic. Not seeing a lot in terms of things like inflammatory bowel disease. Again, not a lot of very specific other things other than I think things that are related to muscle tone within the intestine. This is what I meant in terms of surgeries. There are a few different, I think of them as mostly common childhood surgeries that we do, ear tubes, adenoids, sometimes G-tubes that are necessary, but thankfully not a huge number of other things, hernia repairs occasionally, but not a lot of, like I said, major surgery that require lots of long hospitalizations or long times in the intensive care unit for recovery. So that's the good news. Finally, I'm just going to go through quickly a couple different medications. The reason I list medications are these are conditions that for me feel like they're serious enough that someone's going to use medication. So that's a good factor to see how serious it is. Within this, I'm not going to go over epilepsy. You've heard about that already. But besides that, sleep can be an issue for some individuals, again, related to the brain and behavior. Antidepressants, not always just for depression, but around other behavioral issues. We're seeing that as well. And then, as I said, different things in terms of gastrointestinal issues. For this is just a sort of survey in terms of different medications used for epilepsy. Keppra frequently used, but also many other medications used. And I'm not endorsing any one of these, but just as simply to say what medications have been used. Finally, you've already seen this in terms of the Vineland data, but at least from the Simon Searchlight data from the Vineland, I would say the kids have been very stable in the sense that we haven't been seeing regression. We are seeing improvement, but it's relatively slow improvement over time and a lot of stabilization at some point. But again, there is some gain to be had. And one of the things that I'm waiting to see, but I do think as we have some new technologies in terms of expressive language, not simply saying words as words coming out of the mouth, but other ways of being able to express themselves. I do think the generation coming up who have more use of ACCs and other communication devices, I do think we're going to be able to have more progress in terms of communication. So just in summary, to put all of these things together, the good news is there's no bad news from coming out of this, that really what we're thinking about are things that we know. We're not having surprises. I think we're pointed in the right direction in terms of the clinical trial, the clinical trial readiness, the measures that we're looking at going forward. I'll just simply plant the seed that, and you've seen it already, that I do worry about a medication or something we might use, excuse me, in a trial, that our measures are not the best measures, that I do think there's a possibility that we might see something that would show us an improvement, but we don't necessarily have really good measures to be able to be sensitive enough to small enough amounts of change. And so I'll just plant the seed that as you all, as a community, are going into clinical trials, you all, as Dr. Mom, Dr. Dad, Dr. Grandpa, Dr. Grandma, whoever you are within the unit, the family, you actually are first line in terms of observers. And you may be seeing things, and you may actually be able to help collectively for us to see what's starting to move the needle in terms of a treatment. And so I urge you, as much as you can, take notes in terms of what you're seeing, try and keep track and share that with your study teams and your doctors as you're doing this, because what we predict sometimes might change in a trial, at least this is my experience, is I'm not always right. And I'm happy to be able to say that out loud, but when you do it, you do need to be a careful observer, because we need to collectively be able to learn to move this forward. So thank you. Good to be here.
All right. I think we do have time for a couple of questions. If anybody has anything for Dr. Chung. All right. Job well done. Thank you. Okay, our next presentation is going to be about understanding STXBP1 genetic variations, and Katie Angione from Colorado Children's is going to help us with that. Understanding our kids' genetic reports and their variants and mutations can be difficult, and so Katie's going to make that all simple for us.
All right, thanks everyone for joining. I've had the pleasure of meeting many of you in clinic and also this morning, so hopefully this will be kind of a back to basics review for a lot of people, but we'll go for it. All right, so objectives today are just to review some basic genetic concepts and terminology, go through kind of a long list of different terms that you might have come across in your children's genetic reports. discuss different types of genetic changes and kind of the differences between a benign versus a pathogenic variant, as well as uncertain variants, which many of you may have encountered. And then discuss how this all relates to STXBP1 specifically and its relevance to therapies either now or in the future. So this is kind of the basic diagram that we always put up here when we're talking about genetics. So we all have our cells that contain our genetic information in the form of chromosomes. The chromosomes are basically packages of DNA that are very tightly wound. And then our genes are just segments of DNA that give our bodies instructions to make different proteins that have functions in the body. A protein is made up of something called amino acids, which are just the building blocks that make that protein. And proteins can have lots of different functions. You know that STXBP1 is very important in the brain. And so that's kind of, we go from genes to RNA to proteins. So a gene, again, is just a segment of DNA that codes for a protein. We have 2 copies of most genes. Those are called alleles. So for STXBP1, we each have 2 copies of that gene. And for your children and family members who have STXBP1, they have 1 copy with the mutation and 1 that is typical. So genetic counselors love a good analogy. So the one that I often use is that the genes are kind of like recipes. They're instructions that tell our bodies how to make something. And then the protein is kind of that final product. So the protein is what we are working towards, what the gene is telling our body how to create. And then the amino acids, again, are the building blocks or the ingredients that make up that final product. The genes are made up of different letters, and that code tells our body how to build proteins, again, made up of amino acids. And the sequence of those amino acids is what determines the 3D structure of that protein as well as its specific functions within the body. The proteins can do things like provide structure and support to different body systems, transport materials, protect the body from viruses, and carry out all the chemical reactions that are needed for our bodies to work the way they're supposed to. So a genetic variant, which can also be referred to as a mutation or change, is any alteration in the DNA sequence that's different from what's seen in the majority of people. So there's no perfect gene, there's no correct version of STXBP1 or any other gene. We all have variations, that's why we're not exact copies of each other, we're not all just clones. We have different versions of what is normal or what's typical. So a variant can be benign, meaning that it doesn't cause any problems, it's just a version of normal. It can be pathogenic, meaning disease-causing, or it can be somewhere in the middle, so of uncertain significance. So many of you who've had panel testing or exome testing might have also encountered variants of uncertain significance. When we see those, we try to take our clinical interpretation into account and determine whether that variant is potentially contributing to your child's symptoms or if it's just a normal variation of that gene. So pathogenic or disease-causing variants are what we usually mean when we use the word mutation. So you might see those 2 terms used interchangeably. There are many different types of mutations. So there's going to be a long list here, but I will kind of point to missense and truncations, which is what we see for most pathogenic STXBP1 variants. We know that missense mutations account for about 50%, and then those pathogenic truncations account for somewhere around 25, maybe a little bit less than that. There also are, and we'll go through some of these specifics, but we also see changes called nonsense mutations that cause the body to kind of stop short. There is kind of a stop signal that the body uses within the DNA code to tell the body when it's done building a protein. So sometimes you can see a single letter change causing one of those stop signals to come up before it should. We can see insertions, deletions, or duplications within the gene. So not to be confused with a deletion or duplication of the entire gene, but sometimes we see single letters or small strand of letters within a gene that are added or missing. So this is where my fun little animations come into play. If those of you who were here a couple of years ago have probably seen a version of these slides before. This is just kind of my way of illustrating how the body reads our DNA and how we can see different changes or mutations happening. So the body has a very specific way of reading the DNA code. It can only read 3 letters at a time. So you can imagine a sentence made up of 3-letter words, like the cat ate the rat. One example of a missense mutation might be if you change 1 letter, and then you end up with the cat ate the bat. So it is a sentence that still makes sense, but it is a very different meaning, especially if you're the rat. So we can see certain sense mutations that still maintain some sort of meaningful sentence, but then you can see others where you might change a different letter and end up with something that really doesn't make sense. So the car ate the rat. Because we're in Colorado, of course, it's a Subaru. We can see something called silent mutations or synonymous variants. This is something that oftentimes does not cause problems, but sometimes it does. And that's because there's sort of another level of control on our genes called epigenetics. We won't get into that. It can be very complicated. But most of the time, silent or synonymous mutations do not cause problems, but there are definitely exceptions to that rule, like there are for everything in genetics. A nonsense mutation would be where, again, there is that stop signal that's introduced early, and it stops that sentence or gene short. So this kind of leaves us with an incomplete sentence or an incomplete version of that protein where there is a piece that's missing. So you end up with the sentence, the cat doesn't really tell you anything. You can't really make sense of what that sentence was intended to be. So that's sort of what we see with nonsense mutations with proteins that end up kind of being these incomplete versions of the protein that the body does not know what to do with. We can see insertions. So if there is a complete new word added to that sentence, it changes things a lot. We've got a very different situation now. So sometimes we can have an insertion that gets added into a gene and changes that final protein product to the extent that it works differently from how it's supposed to. We can also see deletions or duplications that can confuse the meaning of that sentence or gene. But if one of those insertions, deletions, anything happens that shifts that 3 at a time reading frame, you end up with kind of this jumble that doesn't make sense anymore. And that's where we can often see early truncations where there's one of those stop signals that's introduced too early. So usually when that happens, the body is creating this version of the protein that it doesn't really recognize and oftentimes ends up recycling those materials and not being able to use that protein in any meaningful way. So how this all relates to STXBP1. STXBP1 is located on chromosome 9, which is one of the autosomes, which means it's not one of the sex chromosomes, X and Y, which comes into play for inheritance. We see that chromosomes, genes that are on chromosomes X and Y can behave differently when it comes to inheritance. And this is why we see STXBP1 pretty equally in males and females. So the STXBP1 protein, as we all know, is very highly expressed in neurons. It regulates the formation of a protein complex that allows for vesicle fusion. And that's necessary for different neurotransmitters to be released in the brain, which is important for all types of different functions that the brain has. So any change in those neurotransmitter levels can lead to uncontrolled activation or excitation of neurons, and that's where we see seizures in this population. And then it also has a role in positioning and growth of neurons during brain development, so things that are very important for memory and learning and all the functions that our brain needs to carry out. So this is an autosomal dominant inherited disease. When we use the word inherited in genetics, it does not necessarily mean that this came from somewhere. We know that in most cases, it's a de novo or brand new change in the child, and it's not something that was passed down from parents. There are exceptions to that, which I will get to in a minute. But we know that it just takes one mutation and one allele to cause the symptoms of STXBP1. So there are rare cases in which a pathogenic variant may be inherited from a healthy parent who might have either somatic or germline mosaicism. This has been seen in very infrequent cases in the STXBP1 population. It is rare, but again, in genetics, we never say 0% for anything. So this is part of why we always recommend parental testing, just to have a more clear confirmation of whether there is a risk of having recurrence in future pregnancies. We know that for siblings or other family members who are healthy and don't have any apparent symptoms of STXBP1, the recurrence risk for them and for their future children is very low. So a little bit more about mosaicism. Mosaicism in genetics is very similar to mosaic tiles like this, which basically just means that people have more than one population of cells. So they have cells that are typical, that have a normal copies of STXBP1, and then they have other populations of cells that might have a mutation. And that can sometimes vary from one tissue to another. So what we see in a blood sample or a cheek swab sample might be different than what is expressed in the brain. So just a kind of more detailed illustration of what this looks like in cells. When there are cells dividing, if there is a cell that has a mutation, all the cells that arise from that will also have that same mutation. So that's how we can see levels of mosaicism over time. So again, most of the time, this is a brand new change in the child. It's not something that's inherited from mom or dad. These types of mutations are something that happen completely randomly. So it's kind of a product of just our normal biology, is that there are always new changes introduced. That's why we have evolution. That's why we are able to adapt to different things over time. So it's a normal process that sometimes just introduces a change in a very important place within STXBP1. Our body has a spell check that helps to correct those changes when they happen, but it's not perfect. So sometimes we see completely randomly these changes happen. When I talk to families, I always try to really drive this point home because we want everyone to understand that this is not something that happens because of exposures during pregnancy. It's not because you had a glass of wine before you knew you were pregnant. Nothing like that is what causes changes in STXBP1. They really are truly random in the vast majority of cases. So again, in very rare cases, the mutation may have been inherited from a healthy parent. We know that there's been at least one family in the literature where germline mosaicism was suspected. So that's where there are egg cells or sperm cells that have a mutation that are not present within the rest of the parent's body. And in those cases, the parents might actually have normal genetic testing. If they're included in trio testing, exome testing, we might not identify a change there because we're not looking at the egg cells, we're not looking at the sperm cells. And even if it is kind of a low-level mosaicism in other parts of the body, it's not something that's always detectable on genetic testing for parents. So just something to keep in mind, anyone who's considering expanding your family, it's always helpful to speak with a genetic counselor, either within one of our clinics or a prenatal counselor who can talk through these things in more detail. I just want to quickly walk through what a test report might look like. So this is just an example from an epilepsy panel. You can see the name of the gene, of course. You guys are all very familiar with that. And then the letters and numbers in the middle there are the coding change and the protein change. So the C stands for coding, it's a change within the DNA, and the P stands for protein, which is the change in the amino acid or the protein. The numbers within those just kind of say the address of where that change lives within the gene. And then for this one in particular, you see the FS*8, so that's how the labs delineate a frame shift mutation. And the 8 just means that it's ending the protein early 8 amino acids later. So we have a lot of jargon in genetics, and it's kind of its own language. So it's always nice to have someone kind of walk through what all of those letters and numbers mean. A test report will include details about why a variant was classified a certain way, whether it's been seen before. STXBP1 has some variants that are recurrent, but many families have their own unique change. That doesn't mean that it's worse or that it's bad to have your own unique variant. There's just certain areas within the gene that are kind of mutational hotspots and are more likely to have changes, and some that are present in only one family in the literature. So understanding your or your child's specific genetic variant or type of variant can be important in determining eligibility for certain mechanisms of disease-modifying therapies. You guys do not need to worry about this for the current gene therapy because that is adding an extra copy of the gene. It's not dependent on the exact mechanism of the mutation that your child has. But for some therapies, it does matter what type of mutation there is, so it is helpful to know. So if there are additional therapies that become available in the future, it might be relevant to know exactly what type of mutation your child has, and that might help determine whether they qualify for that therapy. So just some online resources that I think many of you are probably familiar with, but always good to have. references. And thank you to STXBP1 for inviting me to speak, and always to my Children's Colorado team for being awesome and being here. And thank you guys all for joining.
Okay, I think we have time for one question, if somebody has a good one.
I'm also happy. I put my email address up there for a reason, so if anyone has specific questions, please don't hesitate to reach out. I'm always happy to walk through specific reports.
Perfect. Thank you, Katie. Okay, now we're going to have another one of our family stories. So I'm going to invite Jennifer Wilson and her family.
Well, thank you. I'm excited for this opportunity to talk about Niley. I might need the 2-minute warning. I didn't really time this out exactly. And sorry for the change of order. My son is having so much fun meeting new friends with the other siblings. We did not know where he was at when we were supposed to go on stage. All right, so next slide. A little about Niley. So Niley, you know, the first word I use to describe her is hilarious. Right on time, right on time. She really uses laughter to connect with people, and kind of think Jack Black meets some twerking type of comedy. And I know for me and others, it's the best laugh you've had all day or maybe in a while. So she has that real ability to bring joy to people. She's also very loving and sweet. Through ABA, I'm really glad we got the autism diagnosis and got into ABA therapy. They work their magic. She doesn't totally understand personal boundaries, so a lot of times she was approaching people like this, so they kind of taught her a little bit of space and this side thing, and so she uses that to show affection. And then also, I'm the oldest of 6 girls, so my 2 youngest sisters have had 7 babies in the last 6 years. So those poor first babies were getting a lot of jabs in the eye and mouth. But now she's like really gentle with them, and I know my sisters appreciate that too. And as you can tell from this slide, she loves gorillas. That's her obsession right now. And we have a membership to the zoo. We go almost every weekend. And if you try to talk to her, you know, like, okay, let's go do this. First, zoo, gorilla. You say gorilla? So it is nonstop. And our massage therapist, or her massage therapist I'll talk about, he knew someone who worked at the zoo, so we got to go backstage and meet Cal. And he did like all this stuff for us. He's so amazing. So she really loves Cal and Gunther. And other similar things, music, water, pacing, and mirrors. So next slide. So the pacing is hard for school. She's going to be a freshman this year. So school has been one of the hardest things. I'm optimistic for Centaurus. They've got a lot of great support there. She'll be in the autism classroom. She'll have a para, so she'll have 4 different paras, 1 for each class, and she'll go half days. Usually she's a little over it by the afternoon. So, you know, I think middle school's hard in general, but she spent the last 2 years in a classroom totally by herself with paras who, you know, I don't think really got her. And, you know, near the end of the year, one of them made a comment, she acts like a toddler. And I'm like, yeah, that sounds about right. And they weren't changing her. So we pulled her out of school a little early, and she didn't get to do her continuation, graduation. But so school's hard, and I know they're not getting the best version of her, because as we do the STAR study, Andrea will tell you we had to spread it over 2 days because just being in one room is really hard for her, and she was ready to tear down those walls. But Children's has been great and accommodating, helping her out, and we do that across a couple days. So yeah, as a teenager now, so she'll be 15 in September, and she's showing some teenager tendencies. She wants some privacy, so she'll go to her room and shut the door. And the first few times, I'm like, Natalie, okay? And she kind of pushed me, said, bye. I got the hint. And then she's also emotional. I think a lot of our kids, like even with pain, she just doesn't show emotion or crying. And just really this last year, she loves her Nana and Papa. She knows directions, so we were kind of, I had to be careful because we were going there the next day, but when we went to the store and I didn't turn the way she thought we were going, she just started sobbing and just really upset. But that's a huge inch-stone, I would say, just being able to have that awareness. So this next section, so this is her massage therapist, and kind of this whole thing, I'm the state expert for Colorado. Thanks. I'm still learning because Colorado offers so much. So I've been through kind of the initial 2 years. I'm originally from here. She's born and raised in Michigan, so we're back 2 years now. So if you have any questions, let me know, but I'm also interested to learn from other Colorado families because there's a lot of opportunities here, a lot of support here. So one thing is massage therapy with their CES waiver. That's something I would never think of doing. And it's been so great because the first, he has experience with other kids as well, like her, but, she would barely sit for 5 minutes. Thanks, babe. And you see Chris? And he said, you'll see. And the last few weeks, she's done, she's done 40 minutes of sitting and getting a massage. And I'll just kind of share his wisdom. He views it as like the more time you spend in your parasympathetic nervous system, mood, digestion, behaviors, sleep all improve. So for him, it's just like 1 extra minute there is a victory. And so that's how I kind of try to view it as much as we can, getting her in that calm state. One other thing, I think I'll talk about that on the next page. Yeah, go ahead. So her medical history. So she started having seizures at 3 days old. I ended up having emergency C-section. So we were still in the hospital, and you can see there, that's pictures of her in the NICU. They happened to be doing a photo shoot, so we got some pictures taken. She was on IV phenobarb for 7 days, which totally stopped the seizures. But then, unfortunately, it was like the day we were leaving, they're like, we're going to switch to liquids. And so you're trying to figure out how to even get an infant to take the meds and then send us home. And they're like, we don't know the right dose. So, of course, the seizures came roaring back. So it was like daily. Then we got a second opinion, got her on some Keppra, started slowly going to weekly, weaned her off the phenobarb. And because we didn't have a diagnosis, they kind of for years were just like, the phenobarb can lower IQ, or that's the seizures, so she'll outgrow this. Eventually, especially when she started getting to school, we learned that that was not the case. But she is one of those kids, when puberty came back, or when she hit puberty, the seizures did come back. She didn't have any seizures from 5 to 10. We were off meds for 2 years, but then she started her period December 2020, and then we actually got COVID April 2021. So I don't know if it was a double whammy, but the month after she had COVID, her seizures came back. And that was a really rough year, but I know she's lucky because it takes some time, but when we find the right combination of meds, they're under control. And so she's 3 years, no seizures now. So, yeah. So she was diagnosed at about 6.5 years old. And so this was all in Michigan, but it was just very rough to get therapies. We practically had a beg. She was 1 year and 3 months by the time She got PT. You can see there in the middle picture, she's about 8 months there, and she's really struggling to sit up. And then the next picture is her at 3 years, so she still needed help standing up. She did end up taking her first steps around 3 years, probably walking by 4. But so we had to work hard to get therapy, and then also with the diagnosis, this has kind of been mentioned. 1 minute, okay. I mean, we were so relieved. First, the mom guilt, right? Like I had dental work during my pregnancy, and then her first seizure was after the Hep B vaccine, so we spent years thinking she had a vaccine injury, and we did chelation therapy and all this alternative Googling, and so that was difficult, and I can imagine, like, I see the kids getting diagnosed early, and that must be very, very difficult, but I think it does open up the therapies that are needed. And then I guess I wanted to say this too, just finding this community. I remember when we were searching, and I can't. Yeah, I can't imagine still being on Google, trying to figure out what's going on. And so the sport's been great, the learning's been great, but we also know with that comes loss too in our community. So when I was pregnant with him, a couple teenage girls, we lost them, and I had to kind of step away from the group and get therapy. And I was terrified of her becoming a teenager. But she's almost 15. The seizures came back, but she hasn't lost any skills, and we got them under control. And so, I guess my message is don't be too afraid. I think it'll be okay. And last page, I just want to thank my family. My mom and dad are here. We have a great support system, and I think the really important thing is her forming relationships. And that's what matters the most. And I think a lot of these skills that we tried to, get therapy for and stuff help open up that communication and ability to interact with the world. And, just the bonds she forms, I think, are the most important things for us. And she's got 2 great siblings. Her and her little brother fight a lot. It's so funny. And then Nora, I'll end with this. Even when Nora just started talking, I remember her being a little toddler, just being like, "Where's Niley?" Because it's me, she's always pacing. "Where's Niley? What's Niley doing?" And so she's just always been super aware and helpful. And she wrote a paper in school. She loves being the middle child because she can eat hard candy when these 2 can't, and because she's in charge. So, thank you, everyone. I know.
All right, thank you, Jennifer and Niley and the rest of the Wilson team. That was great. All right, we're going to have one more talk before we take another break. So you guys have heard a lot of results of research in the presentations today, and Charlene's going to hop up here, and she's going to talk about some of the studies that we are prioritizing here at the foundation and that we think will help gather insights for the community. So I'll turn it over to Charlene.
I'm back. So I hopefully am going to just give you a short few minutes to talk about the studies you should participate in. I know that, and if you, I can go to the next slide. I have the power. So we've been talking a lot over the last couple of years about clinical trial readiness. And I know that maybe it feels like we're drilling this in to everyone's heads, but a lot of these, even for me, after learning about this for 5 years or so, some of these terms are still a little hard to, kind of completely grok. So wanted to, go through this in a little bit. So clinical trial readiness, all the work that we are doing with gathering data from patients is really around defining what we want to measure and what we can actually measure in clinical trials so that we know that a future therapy is working. So, and some of this discussion has happened a little bit today. You've seen this disease concept model on the left that was developed a couple of years ago, developed and published a couple of years ago on what our patients and families care about in terms of the symptoms that impact them. And we've communicated that to the FDA in our patient-focused drug development meeting in 2023 as well. So this is what we would like to measure. And now, how do we measure that? What can we actually measure? And this is a really busy slide, but what you'll see is the slide that Sarah presented this morning in terms of the protocol that we're using for the STAR study, as well as the measures that you see in the Simon Searchlight study and other studies, RareX. These are all efforts to get at these different areas. So this is also why we're asking you to give biosamples, because getting those samples enables us to look at biomarkers and ways that we can, hopefully identify whether therapies are working. So I wasn't expecting that, but okay, I showed this morning, but I wanted to just emphasize this around the speed that we're trying to work, because for better or worse, a lot of other disorders have actually had even decades of data collected. I was I went to an Angelman syndrome conference a few weeks ago. They have more than 20 years of natural history data. We have two of clinical longitudinal data. And so because of the fact that we're trying to work so fast to compress time, that's the reason why what my next slide is going to talk about is the studies we want you to participate in, which is going to be more than one. And I know that this is this is not my original one. Sorry, I did actually license a version of this. So this is, I know, how we all feel at in the morning. It's been a long night. Maybe our kid or kids have been up, and we're just really working to try to make it through the day. And now here I am up on this stage asking you to complete another survey or participate in another study. And you probably feel a little bit like this. So what I want to just emphasize to you is that it really is, every single piece of data that you contribute is valuable. It really helps us to build this picture of what we want to measure as well as what we can measure. So you may have seen this. It's our Get Ready checklist, and you can see the items here on the Get Ready checklist. It is very focused on supporting the STAR study. So I'm going to say that up front because there are additional studies that we would like you to participate in. This is, if you can do all of these things, and especially since we're here at the conference, do the biospecimen collection, it will really help us to advance our understanding of endpoints for STXBP1. I want to bridge the STAR study to additional studies because Wendy, who I think covered, a year worth of information in about 20 minutes. I'm always impressed, Wendy, about how you're able to break it down and make so much data understandable. But I wanted to show this, and Sarah Ruggiero showed this morning. This is how we're actually bringing data from multiple studies together. So I want to emphasize this because, no study like STAR, has 146 participants, which is amazing, but it's only 146 participants. And so if we're able to augment that data with data from other studies, we can really start to build and kind of cobble together some of the pieces, that are really needed during this critical time that we're developing clinical trial readiness. So what you can see on the left is it's actually a combination of data from Simon Searchlight and data from RareX for Vineland. And those gray dots in the background are all from Simon Searchlight for other disorders. So that's actually pretty interesting because you can see the comparison of STXers as compared to other neurodevelopmental disorders. Here's just some data that's coming out of RareX. So RareX is a standalone registry for symptom data and other studies. We're also using it for our natural history study. So when you go to your site, they will also tell you that you need to complete these surveys within RareX, and that's for the patient-reported or caregiver-reported outcomes. And so here is one example of some data in terms of behavior as well as impact on different aspects of the CNS or the brain and the CNS. And then I wanted to also just highlight Citizen. So we haven't talked about Citizen probably in a year, and it's not because we're not partnering with them. We're continuing to partner with them. And I wanted to emphasize that Citizen can be helpful for two reasons. One of them is that they gather medical records from different providers that you've seen. So when it's very easy to sign up, and like 5 minutes, and you just sign up and then you add your medical providers, and they will pull and structure all of those medical records. There's a benefit from a research standpoint, but there's also a benefit from a family standpoint. When we did our first visit at Colorado Children's, I just gave them a link to a provider link to my Citizen record for Juno, and I did not have to request or bring any other medical records. It was pretty fantastic. Yes, very easy. And then they structure those medical records, they pull and structure those medical records to support additional research for STXBP1 and other disorders. So it's a really great benefit today from a clinical care standpoint, and also tomorrow. You can also get your genetic report from them. So since I know that that's been difficult to get for some people. Okay, so I think these are my last couple slides, but biomarker collection, or biospecimen collection. I know that Jim mentioned this morning, but wanted to just break it out that there are 3 things that you can do: give blood, do the nitrous oxide measurement, and give a nasal swab. These will be used for different types of studies, but all looking at is there data that we can get from these different types of specimens to help us to advance research? Okay, for those of you who are online, if you're not able to give today, there are several other conferences that are happening for the balance of the year, and Combined Brain is our fantastic partner who is doing the data collection and manages the biorepository for us. Okay. If you have any questions, please email research@stxbp1disorders.org. This is actually the area that Jackie Steinberg manages for us, but she has been running in and out to manage all of the research that's been happening here, so I was stepping in for her. So, okay, I will leave it there, and if anybody has any questions, we can probably take one. We can take one. Oh, okay. So there's a question about Citizen, and since it has rebranded, if you need to do anything new. So you don't need to do anything new. You don't need to do anything new because of the rebranding. If you have new providers, you do need to go in, and you should just do that on a regular basis. Like definitely when you have a new provider, but if you kind of do go in once a year, then you'll make sure that you're up to date, because then they'll go and get those medical records from the new providers as well. They will keep going and getting your medical records for the other providers when they do a data refresh. You always need to give blood. No, seriously, there is actually a reason for it. We are very interested in longitudinal data, and we've actually been asked for longitudinal data. So to be able to see, do those biomarkers change over time? Do those things in your blood change over time? Great. Am I going to let people go to break, or?
I think so.
Yes.
Just a couple things real quick before we go to break. So if you have siblings, I don't know if there's any siblings in the room, but if you have siblings with you, we do have somebody doing a little paint party in the sibling activity room, so they can go in there and do a little painting. I would love for them to paint some rocks, because we're going to use them as decorations this evening. So if you want to encourage your kids to go in there, paint some rocks, even your STXers can go in there and paint some rocks. And then the other thing, I'm going to be putting these on the tables, so when you come back, you'll see these. But this afternoon, totally optional. But our one speaker that we'll have tomorrow, Amanda Griffith-Atkins, is offering a session for families, kind of like a processing session after today. If you guys just want to sit and chat, talk some things through. She's a licensed counselor, happy to do that for you guys. I'll put these on the tables. One more thing. That's it. What else you got?
I was just informed that the nitrous oxide machine did not arrive. So you have two things. You have two things to go and give samples for.
I did remove that sign. Yes, we don't have the nitric oxide. Okay, so yes, please participate in the research, and then we'll see you back. What time? How long? 2:50. See you back at 2:50. You can find these on the tables when you come back. Okay? Thanks, guys.